Almost 2 o'clock. Just let the last few people trickle in, but it looks like we have a number of folks here, which is fabulous. So it's now 2 o'clock. Welcome to the Population Genetics and Molecular Evolution section of the virtual evolution meeting today, Friday. Very excited to introduce our first speaker, Moanfang Ni, who will be explaining how gene regulatory network structure helps us understand how polygenic phenotypes adapt. Thank you very much.
Hi, thank you for the introduction. So, as most of you may know, polygenic phenotypes have a big role in adaptation in many organisms. So it's the case, for example, in plants for adaptation to latitude or altitude with flowering time, in Drosophila for adaptation to cold, in most animals for adaptation to the pathogenic environment, and even adult size in humans can be under sexual selection. So what's exactly a polygenic trait? If you look at it from the standpoint of the quantitative genetics, it's a trait that's determined by several independent loci. So that has been the view for a long time. And then there has been new results from systems biology and functional genomics that have painted a much more complex picture, where those independent loci, they code for genes or they regulate genes that belong to processes. Those processes, they interact with each other. And some combination of those processes code for traits. And what we call a polygenic trait is sometimes a combination of a set of correlated traits. So in this picture, there is a lot of pleiotropy. And it might raise the question, how is it possible for such complex phenotype with such a level of pleiotropy to adapt? And one of the interesting things that has been also brought by system biology is the representation of what's happening at the molecular level as interaction in a network. So this is even more interesting because it's at this level, biologically, most often that the environmental input is registered. So my bet here was that by digging deeper in the molecular structure of those processes, in particular in the gene regulatory networks that regulate those polygenic traits, we could better understand how they adapt. So in this presentation, I will address two main questions. So in terms of gene regulatory networks, how is polygenic trait heritability spread within those regulatory networks? And is there a link between the network topology, the structure of the network, and signature of selection for polygenic adaptation? So the first part has been done by Catherine Stone, a bachelor student that was in the Quackenbush lab when I was a postdoc. And the whole rationale of this study is that when you take SNPs associated to polygenic trait in genome-wide association studies, only a few of them are exonic. The rest is mainly intronic, may fall within promoters or within intergenic region. So those cis-regulatory elements, intergenic elements, promoters, and sometimes regulatory elements within introns, they contain most of the trait-associated SNPs, and they play an outsized role into this polygenic trait heritability. So the question was, how do we access them in terms of representation of molecular function? And one of the oldest and most efficient way to do that is to look at expression quantitative trait loci. So the great thing with the human model is that there are many datasets available, and one of the biggest of them in terms of mutation and gene expression level is GTEx. From this dataset that has more than 50 different tissues for 800 individuals, I extracted 29 tissues that had between 200 and 706 individuals for which we had both genotypes and RNA-seq data. And from those data, we extracted expression quantitative trait loci, that is SNPs that are associated to gene expression level. So for example, here the A allele is associated to a higher level of expression than the G allele, in cis on the left and in trans on the right. So we did cis and trans EQTL detection for all of those tissues. And then the question was, how do we make sense of them in terms of regulation, in terms of complexity? And the answer is, well, you can represent them as a network. So you put SNPs on one side, gene expression that they are associated to on the other side, and the edges are weighted depending on the strength of the association that you detected in the EQTL study. And from this network, you can then look for several properties, including modules, that is SNPs that are particularly connected to genes. And you can look at the position of the SNP within those network. So we had one network like that for each tissue. And then the question was, in those network, where is heritability located? So we added GWAS summary statistics to the mix for 10 traits and diseases with a diverse genetic architecture and a wide range of heritability. And using a stratified LD score regression, we looked for places where heritability was enriched within the network. So we did that for each trait across all of the 29 network. And what we found was that, well, in all of the cases, the heritability for each trait was clustered within a few modules, going from 1 to 25% of the modules that were present in those networks. So most of the heritability is clustered in a few modules. And when we looked at which modules, it became very interesting because they were clustered within biologically significant modules. So for example, here you have HDL level heritability. And this was clustered within a module in adipose visceral tissue that was made of genes belonging to the lipoprotein turnover biological process. So we have this picture. I'm sorry. We have this picture, and then the next question was, within the module, where are the heritability in terms of SNPs? So you have several classes of SNPs that you can distinguish. You have global hubs. They are SNPs that are connected to many genes across all of the network. So they are very pleiotropic, involved in many biological functions. Then you have local hubs, core SNPs that are SNPs connected to many genes, but within one module, within one biological process. And then you have what we call leaves in a network, which are SNPs connected to very few genes. And when we looked at where the heritability was, it was enriched in hubs in general, but mainly it was enriched in local hubs that affect many genes within one biological function. So the second part of this talk is going to be about where is the selection signature of polygenic adaptation located within this network. So this work was partly done by 2 students, and we tested for different signature of selection, negative selection, positive selection, and a statistic that could be a signature of polygenic selection. So for negative selection, we looked at GERB scores, which measure how much constrained a site is. So the higher the GERB score, the most constrained. From this, you can extract constrained regions, and we looked at where the SNPs in terms of topology of the SNPs were enriched for a conserved region. And not unsurprisingly, global hubs and local hubs were both enriched in constrained regions. That's kind of logical if you think about it. They affect functions in a big way, potentially, so they tend to fall within functional elements that are constrained. So the question was now, if we want those complex traits to adapt, those polygenic traits to adapt, where do we find that in the network? So I looked for signature of recent strong selection, recent SNP, positive selection, using IHS that compare haplotype length. And I found that there was no clear pattern for recent selective sweeps between global hubs, local hubs, and leaves. So the last things I did was looking for population differentiation. So taking FST, and those population differentiation is often a signature of polygenic adaptation, can be among many different things. And what I found was very interesting, because the local hubs were enriched for higher FST results, so higher population differentiation compared to live, and global hubs were depleted in high FST, both when we compared African and European population and when we compared East Asian and European population. So it means that the local hubs, those SNPs that are articulating biological functions, regulating biological functions, they are enriched for potential polygenic selection signature. So the whole results, it paints a picture where you have heritability clustered in tissue-specific modules and enriched in global and local hubs, but in particular local hubs. And those local hubs, they are also most likely to be target of polygenic selection compared to the other SNPs. So here we have a structuration and a tissue specificity of the EQTL networks that allows to limit the propagation of the mutation effect to one biological function within one tissue. And this scenario opens up a path for polygenic trait to evolve. So that being said, I'd like to thank everybody that participated to this study, my team at GQE Le Moulin, Fanny Pouillet, with whom I am collaborating on the polygenic adaptation question, the student who worked on this topic, and Fred Osterlitz, who also has helped me think about those questions. If you want to know more, you can check these papers that contain the results that I presented today. And also, I'd like to do a bit of advertisement. If you're interested by such questions, there is a postdoc available in my team studying polygenic selection in gene regulatory network. Thank you for your attention.
Thank you very much for an excellent talk. It looks like we do have one question already in the chat. Sisenga, I'm sorry if I mispronounced your name. Would you like to unmute and ask that question? Alternately, I can ask it on your behalf. Not hearing an unmute, so I'll ask the question on the asker's behalf. How did you have data about heritability, they were wondering?
Oh, so I took available data from published studies, mainly the one that were compiled by the Price Lab in Harvard, and he makes them available to other people. So it's really just the summary statistics of the GWAS studies, but for all the SNPs, so we have the whole picture.
Wonderful, thank you. Oh, that's totally fine. No worries that you couldn't unmute. Does anyone else have any questions? Feel free to raise a hand and I'll keep an eye on for raised hands. We have another 2 minutes for question, 1 or 2 minutes for questions.
Hi. I should raise my hand, sorry.
Thank you.
Thanks for the talk. I was wondering, when you link a SNP to the expression of a gene, is it enough to have just one SNP, or do you have to, how many data did you use actually for?
Oh, so in this case, it was genotyping data for about the whole genome, including, so it was sometimes done with genotyping array, assays, I think, but there was also imputation done, so I had access to the full picture, basically, of the genome. And I entered all of the data in the network. So putting a threshold for significance of the association, but then taking also the strength of the association as an information. But it was many, many SNPs, like millions of SNPs at the beginning.
Thank you.
Wonderful. Thanks very much. I think we probably should move on to get ready for our next talk. So if you wouldn't mind stopping sharing your slides.
Okay, thank you very much.
Thank you very much for your talk. Lisa, if you want to go ahead and start screen sharing.
Yes. This is all right?
Yeah, that's perfect.
Okay, guess I'll start then.
I can introduce you briefly if you'd like. But it's 2:15, perfect. So we're going to have a talk by Lisa Metrop, who's going to speak on variation of mutation rates and spectra in the green lineage. So over to you, thanks.
Okay, thank you for your attention and thanks for the opportunity to let me present a bit of my PhD work. So I'm a PhD student at the Oceanographic Observatory of Bagnols-sur-Mer in France, and I'll talk to you about the variation of mutation rates and spectra in the green lineage. So when I say mutation rates, there you go. I mean the spontaneous mutation rate, which is often symbolized by mu, and can be defined as the number of mutations per nucleotide in a genome per cell division. And since mutations are the ultimate source of diversity upon which evolution can act, the mutation rate actually defines the adaptive potential of a species. And besides, it's an important quantifiable parameter that we can use in population genetics and modeling and evolutionary studies. So we distinguish different types of mutations according to size. So single nucleotide mutations, small insertions and deletions, which are no longer than 50 base pairs generally. And then what we call structural mutations, which include very large sequences or even whole chromosomes. And all of these different types of mutations have their own spontaneous mutation rates. But the spontaneous mutation rate being the total mutation rate of all different types of mutations that arise spontaneously within a population, you cannot measure them in natural populations because we estimate that the majority of mutations that arise are either have a deleterious or a neutral effect on the fitness of an individual. And thus they will not be fixed in natural populations and the effect of selection. So there has been a method devised that allows us to have fixation of all types of mutations, including deleterious ones, except for the lethal ones, of course, which cannot be fixed. And this method, which will allow us to measure spontaneous mutation rates, is called a mutation accumulation experiment. So in a mutation accumulation experiment in asexual species, we start with a clonal parent line. And from this line on, we're going to inoculate many different mutation accumulation lines in parallel, which are going to accumulate mutations in an independent manner. And at regular intervals, we're going to submit these mutation accumulation lines to bottlenecks. That's to say, we're going to drastically diminish their population size. And this will have an effect that genetic drift will rise to a level where selection is negligible. And in these mutation accumulation lines, you'll see the fixation of all types of mutations, including deleterious ones, except for, of course, the lethal ones. And so at the end, we can sequence the the genomes of the mutation accumulation lines and see the mutations that have spontaneously arisen. And this will give us a direct estimate of the spontaneous mutation rate. However, mutations being very rare events, you have to keep these experiments going for a really long time with many lines in parallel. So in our lab, we work on the spontaneous mutation rate in phytoplankton, and we generally keep the experiments going for about 200 to 500 days. So then I'll talk about mutation rate and spectra in the green lineage. And just to place the green lineage, here we have the schematic tree of life. Each icon is a species of phytoplankton. And you can see here that we have what we call the green lineage that falls within the Archaeplastodon. And so the green lineage has two important subgroups: green algae or chlorophytes and vascular plants or streptophytes. And within the green lineage, we already have the mutation rates estimated for 20 species, 7 chlorophytes and 30 streptophytes. Here you have a little oversight of the huge variation we have between mutation rates in the species. So you can see that there's actually between all the species within the green lineage, there's 3 orders of magnitude of variation between single nucleotide mutation rates. And we also see that in general, green algae have lower mutation rates than vascular plants, with the exception of these 2 species of duckweed. So this is already very interesting, but we do miss information about evolution of mutation rate as a trait. And we don't have any information on mutation rates in the neighboring groups like glaucophytes or rhodophytes. And we also have no information about more early branching species within the green lineage. So in my PhD, I've looked at the
Sorry, Lisa, I think we lost you. Oh,
Oh, can you hear me?
yes, we can hear you now. Thank you.
Did you miss a lot?
The last thing I heard was we didn't have data on the outgroups or sister groups that you were showing on your slide.
Yeah. Well, just to say that we don't have much data on early branching species within the green lineage either. And Prasinoderma phyta, its phylogenetic position is debated, but either it's split off before the split between chlorophytes and streptophytes, either it's early branching within chlorophytes, but it is it will give us some information about more early branching species within the green lineage. And besides, it has another very interesting feature is that it has a super high GC content in its genome. So about 70% of its genome is made of guanine and cytosine bases, which is huge. And also it's relatively easy to culture and has a small haploid genome of 25 megabase. So we did a mutation accumulation experiment in Prasinoderma coloniale, which allowed me to estimate its mutation rates. So I counted 43 single nucleotide mutations for callable genome of about 21 megabase over 12,544 generations of the species. And this allows me to estimate mutation rate of 1.62 times 10 to the minus 10 single nucleotide mutations per cell, per nucleotide per cell division. And in the same way, we can estimate the small insertion deletion rates, which is 3.4 times 10 to the minus 11 mutations per nucleotide per cell division. And if we look at the single nucleotide mutation rate and we place that in the spectrum I showed you before, we can see that it's really at the lower end of the spectrum within the green lineage. So it's very interesting. It has a super low mutation rate. But besides mutation rates, mutation accumulation experiments also allow us to look at another feature, which is the mutation spectrum. So I looked at a single nucleotide mutation spectrum, which can be defined as the distribution of frequencies of the 6 different types of single nucleotide mutations, which are here on the bottom of the graph. And so here you see the raw count of each event. And you can see that by far the most common single nucleotide mutation in Prasinoderma was a GC to AT transition. And so that means that in absolute, the GC content of its genome is going down. But since the GC content is already so very high, if you look at the mutation rates of A and T bases that's changed to G or C bases, and the mutation rate of G or C to A or T bases, they are not significantly different. And this might seem evident to you or normal, but it's really not. It's very exceptional. In general, we see that G and C bases mutate far quicker than A and T bases, and we call this the AT bias. And it's the case for the majority of bacteria and almost all eukaryotes. So until Prasinoderma, there were only 2 exceptions known to this AT bias, which were Micromonas pusilla, which is another green algae, and Emiliana huxleyi, which is an haptophyte. And all 3 of these species have a very high GC content in their genome. So this is super interesting. And,
Sorry, Lisa, I think you're breaking up again.
Know more about it, but I'll stop here for my results so far. So as take-home message, we have seen that spontaneous mutation rate in Prasinoderma coloniale is very low. The absence of the AT biases is just very atypical. And sorry, can you hear me?
I can hear you now, though it's a little bit wobbly.
Well, anyways, I'm at the end. Hello?
I can hear you, but it's a little bit broken up.
Hello?
Lisa, can you hear us?
Maybe try pausing
Oh,
your
I'm so
video.
sorry.
Oh. Pause the screen share.
Is Lisa still here? Oh, probably she tries to reconnect.
Possibly. Fingers crossed.
Yeah. So
Oh no.
probably Lisa tries to reconnect.
I think so, yeah.
But-
It looks like she's disappeared from the list of participants.
Yeah. Good thing this happened at the end of the talk. But you can already prepare to raise your hand if you have a question when Lisa comes back, or use the Q&A tool for a question, and then Lisa can answer later. Let's wait maybe 2 more minutes. if
That
she
sounds good to
comes
me.
back. Mm.
Don't forget, everyone, also that there is the option to network on the Zoom platform, the Zoom events. And so if you have a question for Lisa and she's not able to rejoin us, you could ask her there as well. I know I had a couple questions.
Otherwise, we can just wait for the next speaker getting ready.
That's good. Give it 1 more minute, but I suspect Lisa may not be able to rejoin. Maria Angela, would you like to start sharing your screen, or perhaps we should switch over from Yanina's to Maria Angela?
Okay. Can you see my screen? I think
We can see your screen.
Okay, good. Thanks.
I'll introduce you in just a minute, if that's all right.
Mm-hmm.
Just in case anyone's frantically switching sections and doesn't want to miss the first minute.
Mm-hmm.
I don't see your screen yet.
Oh, you don't? I do. Interesting.
Ah, now I can see it. Perfect.
Good.
Okay.
Thank you
good.
for checking. We'll give it another 30 seconds and then we'll start up. And I think probably best to start now. So Maria Angela Ianello is going to tell us about double uniparental inheritance of mitochondria and bivalves, which is associated to specific molecular evolution of nuclear genes. Thanks very much for your talk.
Thank you very much. And I would like to thank the organizer and the moderator for this virtual conference. So my name is Mariangela Iannello, and I'm a postdoc at the University of Bologna. And today I'm going to talk about a peculiar mechanism of mitochondrial inheritance. So as you know, let me see. I don't know, I'm not sure if you can see the top of my screen.
What I'm seeing is the title is Strictly Maternal Inheritance.
Okay, perfect. Okay, so as you know, nuclear DNA is inherited from both maternal and paternal lineage, while mitochondrial DNA is inherited usually just from one lineage. And in almost all animals, mitochondrial DNA is inherited from maternal lineage. And this kind of inheritance is called strictly maternal inheritance of mitochondria, or SMI. And it seems it evolved multiple times, and possibly it has different advantages. For example, it limits the spread of endosymbiotic parasites, it reduces the insurgence of lethal mutations, and it reduces the insurgence of genomic incompatibilities between mitochondrial and nuclear genome, because we know that these two genomes need to cooperate. And we have just one stable exception, and we can find the exception in bivalve species. So in this species, we actually have a different mechanism of mitochondrial inheritance, which is called doubly uniparental inheritance of mitochondria, or DUI. And in this species, both maternal and paternal mitochondria are transmitted to the offspring. And you can see here in violet, these are paternal mitochondria carried by the sperm. And after fertilization, we can see a different pattern of paternal mitochondria distribution in embryos. So in male embryos, we can see paternal mitochondria here stained with mitotracker, they are aggregated, while in female embryos, these mitochondria are scattered, dispersed in the embryo. So during the development in females, these paternal mitochondria eventually are degraded or diluted, and we don't find them mostly in female adults. While in males, these aggregated mitochondria eventually end up in the germline blastomeres, so we can mostly find them in the gonads of the males. And this is mostly DUI, but what we know about this kind of inheritance, it was first discovered in 1994, and since then it was shown in around 100 species belonging to 4 orders and 12 superfamilies. And you can see that the distribution of DUI in the bivalve tree is pretty scattered. We know this is a stable mechanism, and this results in having 2 very different mitochondrial and maternal and paternal mitochondrial genomes. So these mitochondrial genomes can reach up to 52% of amino acid equidistance. We know that usually these mitochondrial genomes have additional open reading frames, but actually we don't know most of the things about DUI. So for example, we don't know how and when it originated. So if it originated once in bivalves and eventually got lost in multiple lineages, or we don't know if it originated multiple times. We don't know the implication of having 2 very different mitochondrial genes within the same species on mitonuclear interactions, and we don't know the function of these additional open reading frames. And importantly, we have no idea about the molecular mechanism of doubly uniparental inheritance. And this is mostly because most of the studies so far focused on the study of mitochondrial genomes, and very few studies focused on nuclear genome. And so the aim of this work was to investigate if there are nuclear genes that show a different evolution in DUI species compared to SMI species. And so hopefully to identify genes that may be involved in this kind of inheritance, of mitochondrial inheritance. And hopefully this can help us also to increase our knowledge about genes involved in mitochondrial function in general and in mitochondrial inheritance in general. So these are the species we analyzed. So 36 bivalve species, and 10 of them are DUI species, here involved with this symbol of paternal mitochondria. And you may notice that for getting coding sequences from these species, we use both genomes and transcriptomes. And this is mostly because, unfortunately, for many taxonomic groups, we don't have a lot of genomes available. So we had to combine transcriptomic and genomic data. And so we retrieved coding sequences and we inferred single copy orthogroups for these species. And we ended up with 8,700 single copy orthogroups in at least 15 species and in at least 5 DOI species. And so starting from these orthologs, we calculated the branch length of gene trees calculated on the species tree topology. So the first thing we wanted to test was if there are genes showing a convergent rate of protein evolution in bivalves with doubly uniparental inheritance of mitochondria, and hopefully identify genes that may be involved in this phenotype. And for doing this, we performed a relative evolutionary rate comparisons, and in particular, we use this tool Tracer, which is an implementation of this rare comparison, and this program makes no assumption about the origin of the trait, which is important for us because we don't know it. And this tool identifies genes with convergent rate of protein evolution. And we found, using this tool, that we have 354 genes with a significant constrained evolution in DOI species, and 89 genes with a significant accelerated evolution in DOI species. And of course, we wanted to know if these genes, the number of these genes is higher than expected by chance. And so for doing this, we performed the same analysis using several different random controls. And we plotted the distribution of the genes found with Tracer along with their p-values. So for example, you can see here in gray and in blue, this is the distribution of genes and their p-values in mostly random controls. And in pink here, this is the distribution of p-values in DOI species. And you can see that there is an enrichment of significant genes with constrained evolution in DOI species. So the number of these genes is actually higher than expected by chance. While for accelerated evolution, we don't find this pattern, quite the opposite actually. So the number of these genes is lower than what expected by chance. So we decided to analyze these 354 genes with significant constrained evolution. And to validate these genes, we also asked if these genes show different selective pressures in DOI species compared to SMI species. And so for doing this, we calculated the dN/ds in DOI and SMI species using CodeML. And we found that 206 genes out of that 354 show actually a significant and convergent stronger purifying selection in DOI species compared to SMI species. And here you can see the distribution of the delta, dN/ds, and negative values means that the dN/ds of DOI species is lower than the dN/ds of SMI species. So for these 206 genes, we actually have a constrained rate of protein evolution and a constrained rate of protein evolution and a different dN/ds in DOI species. And we asked if these genes actually have a role in mitochondrial biology, or they may have a role in DOI. So for answering these questions, we performed an evolutionary rate correlation analysis. So this analysis, the point of this analysis is to correlate branch length of different genes. And if the branch length of different genes are correlated, usually it means that these genes have the same evolutionary pattern. And so possibly they're involved in the same functions. So what we did here was to correlate the branch length of the mitochondrial tree with each of our single copy orthogroup genes to find genes that may co-evolve with mitochondria. And we did it for all of our dataset, for all genes from our dataset. And we found 1,100 genes showing a correlation with evolutionary rate correlation with mitochondria. And we saw that these genes actually show a strong geotherms enrichment for mitochondrial functions and localization. These genes are enriched for protein-protein interactions. And actually, most of the genes that we know have a role in mitochondrial biology, we can find them in these genes, in these 1,100 genes. So this is actually, it seems, a good proxy to identify genes that co-evolve, nuclear genes that co-evolve with the mitochondria. And they may have a role in mitochondrial functions. So what about our genes, the genes identified with Structure and validated with CodeML? Okay, we found that these genes are actually enriched for correlation signals with mitochondrial proteins. And so perhaps most of these genes may have a role actually in W uniparental inheritance of mitochondria. And what are these genes even at the end? So we performed a functional annotation of these genes, and you can see that among enriched GO-terms, we don't find a lot of genes involved directly in mitochondrial function, but we can find several GO-terms involved in microtubule organization, for example. Now we know that mitochondria are very dynamic organelles, and their movement and distribution depends on their anchoring on microtubules, thanks to motor proteins. So we are still analyzing this data, but so far we found some interesting genes. For example, we found the most of the genes from this house complex, and this house complex in neurons, it seems it has a role in organizing microtubules. And also we found this gene here, metaxin-1, which also in neurons, this is localized on the mitochondrial outer membrane, and in the neurons it seems that it binds kinesin, and it allows the movement of mitochondria in cells. So as you may notice, most of these studies focus on neurons, and possibly this is because when it comes to migration and studying the movements within cells, most of the study focus on neurons. But it is tempting to think that possibly the same genes may also have a role in the distribution of paternal mitochondria and have a role in this differential pattern in DUI species. But again, we're still analyzing this data. So
it's just FYI.
Well, I'm running out of time.
You have another 2 minutes.
Okay, great, thanks. So our conclusions so far are that the DUI phenotype seems to be reflected at the genomic level in a convergent evolution of a set of nuclear genes. That most of these genes co-evolve with mitochondrial genes, so this validates the possibility that these genes are involved in DUI. And we are discussing that some of these genes may have a role in the transport of mitochondria, and possibly they may have a role in the different segregation of paternal mitochondria in the male embryo. And just very quickly, we would like to validate this data with transcriptomic data. So we are planning to perform a differential expression between embryos with different pattern of mitochondria to see if we can see to validate, hopefully, the results we are getting so far. And I would like to thank all the group, the Evocom group, and in particular, Giovanni Piccirini, who is the co-first author of this work. So thank you very much, and sorry for the late.
Thanks very much for your lovely talk. We have 1 minute for questions if anyone has a question. I do see some applause, which is fabulous. No questions in the question and answer. I might start by asking a question, actually. I'm curious, so you've presented in the beginning some advantages of purely maternal inheritance. What's the advantage in bivalves of the double inheritance? Yeah,
that's a great question. We actually don't know if there are advantages or if there is something, a selfish element that may cause this kind of inheritance. It doesn't seem so far that we have some advantage. So we actually don't know yet.
Wonderful, thank you. I think we'd probably better move on to the next talk. I'm going to stop recording now, just for the info for everyone else. Thanks very much, Shivani. We'll give it about 30 seconds before we start your talk, just to make sure that anyone who wants to trickle in can trickle in. Are you able to put that into full screen by any chance? Perfect. That's grand. Thank you. Just waiting for it to turn. Okay. So our last talk of the session, and thanks very much all of the speakers so far, and thank you for joining us. As well, the audience is from Shivani Selimani, who's going to present repeated adaptation to habitat in the extremely variable and widespread common evening brown butterfly. Go ahead, please.
Oh, thank you. Am I audible?
Yes, we can hear you just
Okay,
fine. Thank you.
Thank you. Sorry. My talk is about geographical distribution of wing pattern trace of a widespread butterfly. Widespread species colonize different regions and experience various habitats across the range.
Sorry, Shivani, we can't see your video. I don't know if you mean to have it turned on.
Oh, sorry.
Nope, that's all right. Thank you. That's perfect. Okay, just wanted to let you know. Go ahead.
Widespread species colonize different regions and experience various habitats across the range. For species that rely on polymorphic cryptic color patterns to avoid the predator's attack, different habitat will select different color patterns. Hence, there will be differences in color patterns between distinct habitats, indicating local adaptation. As you can see in my illustration here, I use snail icons with different color outlines. Snail icon with a black outlines is more likely to fit in a dark green habitat representing forest. Meanwhile, snail icon with a white outline is more likely to fit in a light green habitat representing grassland. And to show that, I have a study by Silverton and others showing differences in color patterns between distinct habitats within population of the polymorphic land snail, Sepia nemorharis. Such local adaptation to habitat is especially evident when a species occur in similarly contrasting habitat in different regions, and there show similar shift in color patterns, indicating that adaptation is repeatable. Just like this study published by Nature Communication showed. Moreover, one region may have low genetic variation and the other may have high genetic variation. A region with low genetic variation, for example, including only one color morph here, which is well fit in grassland, as shown in region 1. Meanwhile, in region 2, due to its high genetic variation compared to region 1, has both phenotypic variation, meaning more fit and less fit. This indicates the relationship between genetic and the phenotypic variation. In region 2, the outline are more cryptic and common. However, this abundance of content-specifics with similar color patterns is a further selection pressure. When the abundance is higher, predators are more likely to form a search image to detect a common cryptic. The individual can benefit from having a rare phenotype. By negative frequency-dependent selection, the fitness of a common phenotype decreases and they become less common. Meanwhile, a rare color-morph individual may increase in number and variation under apostatic selection. And hence, the abundance of regional 2 increases over generations, and some new variations can be even harder to detect. You can refer to review article by Bond 2007 regarding the evolution of polymorphism. Therefore, this suggests that the greater diversity among individual color pattern is associated with increased mean or average abundance. Study by Forsman and others indicate this relationship. Much of snail icons, I know, and I'm done using them. Our study species Melanitis leda, also known as common evening brown, is a widespread colonized or occupy different regions across Africa, Asia, and Oceania. Its habitat ranging from foresty, like tropical foresty, rainforest, grassland, like savannas, is often very abundant, as you can see here. In this photo, many individuals gather at light, but sometimes we get few. The third species also has color variation in many traits, See the background varying from reddish to orangish, grayish to brownish. There are darker blotches that can be present or absent. They differ in size and color. Then there are borders that can be clear, blurry, or absent, as well as have different shapes. It takes about 12 minutes to score all these traits that I've described between individuals within a population. By the way, these photos were taken in a single day at one place in India. I can add another morph here, even more. It's fascinating, isn't it? The larger number of polymorphic variants offer the perfect target to understand the process of evolution of a species, as it provides easy to measure phenotypic traits that can be expected to be associated with genetic lineage, habitat, and species local abundance. So we first predict that the range of color patterns in a region is higher where there is more genetic variation. From a study by Latore, we know that our species originated in Asia. So we may, for example, expect less genetic variation in Africa, and thus also less phenotypic variation. Based on two populations and a few key traits from pictures of butterfly individuals collected from different locations of Africa and India, research shows higher phenotypic variation in India than in Africa. For example, here, a frequency bar chart of the forewing and hindwing border categories in India and Africa individual. India is represented by orange and Africa by light green one. Indian butterfly have significant more abrupt color changes in the border, while African ones tend to have gradual or absent color changes. The frequency bar chart of hind wing blotches show there's higher frequency in the presence of clear black blotches compared to African one. Moreover, the lack of spotted African individual is also noticeable. This pattern may be explained by reduced genetic variation in Africa due to founder effect or low abundance at African sites. We have another hypothesis which says populations show consistently higher frequency of darker phenotype in a more woody habitat, as the brightness in grassland favors higher phenotype, while canopy in forestry favor dark ones in terms of camouflage and predation defense. This graphical representation of this hypothesis is adopted from the study of Mooring and others. And finally, we predict that there is more variation where the species is more abundant. And the strength of a frequency-dependent selection can vary with region due to difference in abundance. To test these three hypotheses, we are now expanding the study to measure a larger number of wing pattern traits using automated image analysis based on machine learning. As I said earlier, it takes about 12 minutes to score all the mentioned traits between individuals within populations. If you take 20,000 photographs times 12 minutes, that will take forever. So machine learning will assist in automation to score wing pattern trait at a global scale. Up to now, we have collected more than 20,000 photographs across the range from different sources, including monthly monitoring, studies, collaborators, online sources, and museums. This work is in progress, so stay tuned. I must thank the National Center for Science, Poland, which funded this project, and my supervisor, Frederick Merman, with a butterfly on his forearm here, Melaniti Slater team at UAM, and other collaborators from different institutes who contribute a lot to this study. And last but not least, we have a call for samples of butterfly. This presentation is a part of our project, which has three main goals. First is to reconstruct a phylogeograph of this species, Melaniti Slater. Second is to compare polymorphic color patterns among regions and habitats. And third goal is to describe seasonal phenotypic plasticity under different climates. We are welcoming contribution in terms of specimen in ethanol, butterfly pictures, and in case of butterfly picture, we are more interested of that we showing undersides. And here, monthly monitoring. We offer authorship funding, we supply traps, grade cards, envelope, among others, and we are inviting collaboration. And we are staggering in Australia for monitoring sampling there. So for more information, please visit our website, our email. This is the email of my supervisor, and this is my email for more information on contribution. Thank you for listening.
Wonderful, Shabani. Thank you very much for your contributed talk. Very interesting. Are there any questions? I'm happy to start us off with one. Do you know anything about the genetic diversity in the populations in Africa versus South and Southeast Asia? Or is there an evidence of a bottleneck from the genome as well?
I think this project will reveal that we don't have such kind of study in the previous. So this is the first study which will tell even if there will be a new subspecies. So this is the first genetic studies, and my part is not genetic per se. I have another student here, in my colleague, another student who is dealing with the genome part. But also we have a collaborator student, PhD student from Queen Mary University, who is dealing with the genome, it's called the Queen Mary University of London, who's dealing with the distribution of gene of the polymorphic of these butterflies. So it's very, very, so stay tuned. I think when the studies is done, to contribute a lot to the knowledge.
Wonderful, thank you. I would be curious personally to know if it was genes that are also implicated in color patterns in other butterfly species. So I will stay tuned. Thank you.
Yeah, and we have more studies with the older study in 1994 by Blakefield and Luther and Blakefield. They studied the interaction of genetic polymorphic and seasonal plasticity. So it's there.
Very cool, thank you.
Yeah, thank you.
Does anyone else have questions for Shabani? Or you can ask in the chat or in the Q&A if you have questions for any of the other speakers as well, of course. I don't see any raised hands. I will keep the Zoom open for another 4 minutes or another 3 minutes now, because if anyone wants to ask questions of Shabani, they should think of questions and ask them. And do, of course, feel free to join us in another room in about half an hour's time. Oh, Parker Hughes has a question. Parker, would you like to unmute yourself?
Yeah,
go ahead.
Really good talk, Shabani. I thought it was pretty interesting. Is there any evidence of like niche partitioning across butterflies of different colors in the same area? Just thinking of like any diet changes to help supplement like carotenoids in their diet, or if they all kind of occupy the same like zone.
From what I know, most of the time, from the rich experience of mine from the field, most of the time is due to microhabitats. So the background is much more correlated with the variation in case of their wing color patterns. So I don't know about the food, hosted plants. No, I don't think of any study, but my experience tells me so about the micro habitats, the background of the habitats.
Excellent. We have another 2 minutes. Does anyone else have questions? Actually, I have a question again, if you don't mind. So these different polymorphic variants, do they cluster by geographic location? You mentioned, for example, in woodier areas, there's darker color, but do you tend to find some polymorphic variants in, for example, Ghana and others in India, or is it sort of randomly distributed?
Yeah, the way it seems that in Asia, there are fascinating polymorphism there, but there are very few polymorphism in Africa. That's what we know. We don't know about Oceania. I think at the end of this study, we'll know for sure. But I've collected, I've said about it, I've collected more than 2,000 photographs myself. Most of them, 80% is me. So I've seen most of the pictures in Asia. particularly India, it's fascinating. A lot of polymorphism for this butterfly. You can just tell this is another species, but for sure it's Melanitis late. Yeah.
Wonderful. Thank you so much. And I think we've come to the end of the session. So thank you to all of the speakers for your wonderful contributions. Very much appreciated. And thank you to the audience for listening as well and for your questions. And I believe we're going to restart a next section at what's 4:00 p.m. for me here in the UK, what would be approximately 11 a.m. for those of you on the East Coast of the US. And feel free to convert to your time zone otherwise. Thanks so much for attending, everyone. Take care. See you soon.
Thank you.